And we are recording.
Great. So hi, everyone. My name is Elizabeth Strychalski, and I lead the Cellular Engineering Group at NIST, the National Institute of Standards and Technology. So today I want to talk a little bit about making measurements in biology and in engineering biology more specifically, but also how to make sure that you're interpreting your measurements, you're finding meaning in those measurements in a way that's appropriate. For all of you PIs who are listening in, you won't hurt my feelings at all if you sign off. I've definitely aimed this talk more at folks who are a little bit earlier in their career, who maybe haven't thought as much about metrology and meaning in engineering biology. So before we get too far, I want to review what it is we're even doing here in the first place. So Build a Cell, we all know because we're part of it, right? We're a community of researchers who have this shared vision of building synthetic cells. And the way that we feel that we need to do this, we value coming together so that we can do something that's bigger than any of us individually. We want to create community. We want to educate each other. And we want to create mentoring and networking opportunities. And that's part of, we're doing all of these here today. And I like the idea of a moonshot for building a cell because it sort of stands in for something that is really, we just feel that it's worth doing. But even though, we're not able to necessarily point to near-term deliverables that give us kind of instant gratification or instant payout. And I think that's the case so far for building synthetic cells. It's something that I think we've been struggling with. And if we can point to what some of those nearer term deliverables are, then maybe we can help focus some of our proposals or some of our work in order to build resources for ourselves and for each other. So that's another conversation. If anybody wants to have that conversation with me, I welcome it. But also, we say moonshot because they were ultimately successful. So maybe someday they'll say, that's your synthetic cell once we've built it. A little bit about me. Again, I am the group leader for the Cellular Engineering Group. You may remember me from such workshops as the Build a Cell workshop number 7 that I led with Jane Romancova, who is giving this seminar next week. And again, she'll be giving much more technical detail on our Cellfree program. Oh, this is where I also have to give the NIST disclaimer. So any of the commercial products or methods that I talk about here today or that we might talk about in the Q&A in no way implies endorsement by NIST. So NIST's mission is, we're one of the oldest national laboratories, which is fun. And our mission is to promote industrial competitiveness and improve the quality of life for Americans through measurements. And I'm showing you here the organizational structure for NIST in case you want to reach out and look for collaborators, look for people who might have equipment you want to use. I happen to live in the material measurement laboratory. This is where a lot of our biometrology lives, but people are doing biomeasurements and building tools for biomeasurements pretty much throughout NIST. I wish I could invite you to our campus. Unfortunately, I can't. But what I'm going to do is share my slides with Kate to post on the website. And I invite you to click through some of these links. I'm showing you pictures of our Newton apple tree that's on our Gaithersburg campus in Maryland. This is the outside of our semiconductor electronics cleanroom fabrication facility. And of course, this is the schematic, which is the crowning achievement of humankind, the redefinition of the SI. If you haven't looked into that, please do. It's really a wonderful accomplishment. So NIST was founded in 1901, and we're part of the Department of Commerce. And you need weights and measures if you're going to agree on trade. So that's why we live there. And the need for NIST and for standards is often dramatized by the story of the Great Baltimore Fire of 1904. So basically what happened was that Baltimore was burning down, and lots of firefighters from the region came to help put out the fire. The problem was they couldn't hook up any of their hoses to the fire hydrants. So the city's burning down around them, and they're all going, oh, darn, right? So afterwards, NIST was called in to create a national standard from the 600 or more standards that they found. And so this is a very sort of visceral and very urgent example, right, of why standards matter. And not all standards will feel this way, but it's a good example. Speaking of standards, if you look at the smaller of the pyramid, so if you go and Google standards or traceability, you're going to find lots of pyramids and lots of triangles. that illustrate different aspects of these concepts. I want to show you one standards pyramid that I stole off the NIST website. And what I like about this pyramid is it really drives home that standards are not dictated from the sky, right? They don't come top down. So NIST is Department of Commerce, we're non-regulatory. So the way standards happen is that we get everybody together. So it's a very bottom up, it's a very grassroots. And we all sit around a table, literally, pre-COVID, and we agree that the field would be best served by standardizing something. It might look like an artifact. It might look like standard protocol. It might look like a lot of different things because there are a lot of different kinds of standards. So we might all be doing assays. You see that bottom block in the pyramid. Then we might come together and agree on some experimental design or interlaboratory testing or control strategies to do better assays. And eventually those might get distilled into something like a standard. But standards take a long time and they take a lot of consensus. So I know some people have fears about standards getting imposed on them. That's really not the case. And I'm happy to talk with you more about that if you like. The other pyramid is more about traceability, and it talks a little bit about some different kinds of standards. So on the bottom, you have process instrumentation, you've got working standards, then you start getting to primary standards. And eventually NIST gets involved, and ultimately you have at the top the SI units in this particular example. So there are lots of sort of interim standards or conditional standards, or those aren't the technical terms, but standards that aren't like, international committee, document enforced standards, whatever, ISO, blah, blah, blah. There are a couple different ones, different standards committees really that we sit on. And every country has their own metrological institute, and that's who all comes together to agree on things like SI units. Are there any questions about any of this? I know I'm going through it really quickly. My purpose here is just kind of say a lot of these words at you so that when you come across them in the future, your subconscious will go, oh, this was important. I should stop and look this up, or I should stop and consider how this might make my measurement better or my experiment better. Okay, great. So as far as biometrology goes, that's what my group focuses on. It's what my division at NIST focuses on. And we've defined this as measuring bio stuff, right? Which we know is hard. So how are we going to take what we know to be the reality of biometrology and then merge that with standards to hopefully get something that's more, experiments, measurements that are more reproducible and more comparable? So we know that measuring bio it's hard. These are small, wet, squishy, dynamic, complex systems. Many of us make measurements in relative units, which makes it comparability more challenging between different experiments in the same lab and between different labs. We know that biology is still moving towards a cultural quantitation. They're not really there yet. And we've been applying tools for measurements that have, in many cases, been taken from physics or chemistry or material science and so on. But I think it's time now, especially now that we have so much facility with starting to build with biology and engineer biology. Maybe we can make entirely new tools based on biology to measure biology. And this is where my group starts to, hopefully we're going to have some impact there. And of course, you want to marry this with, meeting that complexity with cutting edge machine learning, automation, high throughput data, and so on. So making the measurements is hard, interpreting them is hard. Kate and I were talking at the beginning of this seminar about how biology has a reproducibility problem. And all of this comes together then when you try to engineer biology, because you can't really engineer something if you don't have good measurements for it. And all of this matters to all of us, right? Because you need good measurements to publish for your scientific reputation. If you want to start a company or invest in a company, you need information based on good measurements for safety and so on. So measurements matter. And I talked about that reproducibility problem. This is just one example from 2016. Where a survey was done about reproducibility in the different fields. You can see that biology is not looking so good. Biology and shockingly medicine as well. About 50% of the published literature is considered reproducible by folks in the field. So So what are we going to do about it? Well, I was thinking about some things that maybe we as Build-A-Cell can work on. And one of these is we might think about standards for reporting. And this is an example from communications biology for just growing bacteria. And I'm not saying we need to adopt this particular standard. I'm just saying that this paper is a nice example of a way of thinking about part of your experiment or part of your workflow, where you might think about a minimum information standard so that you know if you hand that off to somebody else, all they need is, in this case, this table to be able to reproduce, in this case, bacterial growth. So, I'd love your suggestions for which parts of the workflows around, building synthetic cells or moving towards synthetic cells might benefit from this kind of a reporting standard. I started to make this slide this morning because I realized I had completely left out experimental design. This is a thing, right? If you don't know this is a thing, know this is a thing. Experimental design, sometimes design of experiments, factorial design. I was looking for links on these things, and I kept coming across the NIST ITL Engineering Statistics Handbook. And if you want, if you want to walk down memory lane, what the internet looked like about 20 years ago, this is a great link to follow. But there is good information there. It's a good place to start if you don't know where to go. People take full classes in this stuff. But again, this is something we're borrowing from engineering fields. And so if, as you design your experiments, I think we can go a long way with better experimental design. And, so much of the complexity of biology just feels like this miasma hanging over us, but it's really an opportunity, right? And once we can get better measurements and better experimental design to build our understanding, to build better models, I think that we're going to be pleasantly surprised with how we can build control strategies for engineering with biology, including for building synthetic cells. Not that that helps us right now, but that's my feeling. And I want to mention too that, there's lots of a cultural appropriation we can still do from the engineering fields that we haven't really fully enacted yet. So when I was a program manager at DARPA for the biological control program, one of the things I really wanted to do was bring over this concept of independent verification and validation, IV&V, from other fields to biology. And it's easy to say, hard to do. But I invite you to look over this editorial written by some of the performers on that program, as well as the program manager who took it over. And then this arrow points to it's one of the performer teams that incorporated their IV&V results in their paper. So with this program, I funded 2 teams for every project. There was the main performer team, and then there was the team that was going to reproduce those results. Now, not everybody's going to have those resources, but it's interesting to really bring the idea of reproducibility and give it such a central place in the experimental design and in the workflow. Okay, so we've been talking about reproducibility, about standards, traceability, but what about the actual measurements themselves? So here is your virtual tour of measurements and uncertainty. If you don't know what the Vim and the GUM are, take a look. I've also included links on traceability, examples of different types of standards as you think about what synthetic cells might benefit from in terms of standards. I also have a link to 𝗕𝗴𝗶𝗴𝗆 fishbone diagrams, and I'm going to show you an example of our thought process around measurements and uncertainty that involve making these fishbone diagrams. And really all I want to say here on this slide is that if you find any of this tolerably interesting, please come work at NIST. It takes a certain kind of person to really enjoy this. Now, we should all be using these concepts and using these resources as we make our measurements. Of course, right? We're scientists, we're not animals in the wild. We're like, uncertainty analysis is really what separates us from the wild animals in my mind. So, if you like this stuff, great, let's talk. And I want to mention too that your ability to fantasize unmitigated measurement disasters is actually a key asset, because this is most of what I do all day. So this is an example that I stole from Jane. She's going to talk more about this next week. But if you have a basic workflow for cell-free protein expression, you prepare your cell extract, you put the reaction together, you add your DNA template, you synthesize your protein. Great. So this is my workflow. And then I want to understand, where am I picking up on measurement uncertainty in this? Where might I, where along my workflow might something be happening that would cause me to doubt something about my final result, or give me information about interpreting my final result? So then we can take each one of these steps along the workflow and break it down into sort of sub-steps. And then we could zoom in on each one of these circles now. And in this case, this is for the DNA template prep, and break down, and this is just, Jane and me brainstorming, like, where can it go wrong, right? And so even just for this one step in our workflow, look at all these places where you can pick up measurement uncertainty. It's staggering. No wonder we can't do this reproducibly. So then what we can do is, once we identify where we might be picking up irreproducibility, we can start looking at those things and understand, well, how do we control it better? How much of an effect does this have, right? We can start sort of prioritizing which sub-steps we might need to nail down better to get a more reproducible result. Okay, so every time I look at the slide, I always feel a little tired and defeated, but hopefully it's motivating for you. So if you haven't done this with your workflows, again, I invite you to do so. I think that, certainly I don't know of a better way to diagram out my workflow to try to understand how to improve it. If you know of a better way, please drop it in the chat, share it with us, or send an email out to Build a Cell later. Okay, so before I move on to my group and what we do, are there any questions on any of the things we've been talking about? I see that there are some comments about we need standardization, lots of standardization needs to happen. Translation, liposomes, got it. And I might actually grab the chat after this if nobody minds, and take this back to my group so we can take your suggestions seriously. So just know that you're not typing in vain. Okay, so I'm going to move on then and talk about my group. So the cellular engineering group came out of the engineering biology team, who I know some of you have interacted with in past years. But we're a real thing now. We've got our own funding. It's a great time to be in this field, of course. And our mission is to provide that foundation of measurement assurance to support control and rational design of biological function. Just control the life force, no big deal. I want to show you the people in my group because, I want to ask you all for ideas or for resources for how we can recruit better. We're pretty diverse, but obviously there's always room for improvement. And we're small. I also want to show that we're quite small. So we've got a big job, but we're not a lot of people. And I will mention that we are recruiting. We have 2 funded positions open right now. One in machine learning and one for SynBio wet lab work around genetic sensors. So I'll mention those again at the end of my talk. So I'm going to show you some capability slides, because one of the ways that NIST operates is we don't just take input from you in the form of Zoom chats, right? We want to work with you. We are resourced, and it's our job to help you make better measurements. So if you understand what we can do for you, and you understand how to get in touch with us, which I think my email address is on every slide, so please reach out. Please consider us a resource. So my group is a little bit like lasagna, where we have a lot of deep expertise in different kinds of measurement platforms. And that expertise, of course, we're the same people, never really goes away. And so we try to fold that into what we're bringing to our current focus, which is, engineering biology. So for example, I have fluidics up here. We've got a lot of great micro and nanofluidics experience in the group. Whether we're using it now or not, it is a capability that would be available to helping you with your measurements. As far as biological systems go, you'll notice there's nothing in our mission statement about biological systems, because we really take that physics and engineering approach of looking for generalizable rules or concepts that can guide engineering of these systems. So the systems that are, all these systems are ones that NIST has expertise in. My particular group has expertise in the ones that are not grayed out, although I will mention that we're looking into how we can have an impact in measurements for plants, for engineering plants. So that's really exciting. And this is another way of showing our capabilities. So I did mention, we're automating our laboratories, we're developing different workflows, and then we have some measurement platforms that we just have on hand. So feel free to, again, you'll have these slides, look through them to see if we can help you with something. And with regard to automation, this is what our main automation lab looks like. And this is the Living Measurement Systems Foundry. This is our BioFoundry. And we are part of the BioFoundry Alliance. So, you can reach out to us or reach out to the Global BioFoundry Alliance to find out more about, our lessons learned. We are talking with other folks setting up BioFoundries so that we can teach them about what was hard for us, what went wrong for us, what went right, as we were looking at automating our workflows. We're also very happy to share our automation methods with you so that you don't need to program them from scratch, right? You've already paid for them with your tax dollars. And the reason I show this is because, if you've been around labs that look like this, then you don't necessarily have an appreciation for the fact that the future of biology, arguably looks more like this. This is a picture down the axis of our LMSF, our BioFoundry, starting, you're seeing the wet deck of our Hamilton Star and then our robot arm in the middle. If you're curious, this is all of the hardware that's on the side of this thing and all the things it can do. And if we were walking around this, again, we can't all be there together. So these are just some snapshots of different parts. And we can pull all of the pieces out. So the way that we've designed our Foundry is that when you're running a process on it, you don't sort of lock up all the parts. Somebody can run one of the plate readers when you're using another one for your automated process, which is nice. And we also have it set up in a way that takes advantage of flexible automation. So, companies that have warehouses full of robots are making, 1 million of the same measurements. But what we want to do is we want to make 1 million measurements, but they might all be a little bit different, right? Because you want to go in the lab every day and measure what you want to measure, and maybe in a different way than yesterday, based on what you learned. And part of this also is because what you're not seeing here is on the back end of this, we're working with really great collaborators in machine learning who will hopefully very soon be able to integrate in with our hardware so that we can go around that design, build, test, learn loop for engineering biology in a way where, after the learn, the machine learning actually algorithmically drives that next turn around the design, build, test, learn loop, if that makes sense. I know there are some companies that are claiming to do this. I think that there's still a lot to learn about realizing that kind of workflow. And this is just some more about our labs. We've got flow cytometry, in-house sequencing. In a previous life, we've done a lot of single molecule microscopy. So we do have those microscopes as well still. And we have a separate automation for things like DNA library prep. So that's what our lab looks like. And then as far as what we're doing with all of that automation, it's a lot of genetic sensors towards control engineering, all with the goal of driving towards this precision engineering biology. And what we mean by that is, not just, for example, on-off switches that may be leaky, right? You want to be able to spec out, for example, what genetic sensor you want, and to be able to hit those specs in a quantitative way in a reasonable time frame. And to do that, not just for E. coli, or lab strain, whatever, but for your organism of choice, and you're operating in your environment of choice. So that's our vision. I've got 3 examples of some programs and projects that might be of interest to you folks. One is this is our big genetic sensor project. And what's really interesting here, I'm not going to go through this whole slide, but I want to point out that the idea here is looking at the directed evolution workflow we all know and love, and saying, well, instead of doing selections at the end, why don't we just measure everything as we go? And Drew Tack and David Ross in my group especially, although this has really been a group effort, have made some great progress in that sense. So, this really fits in this mindset of measurements. So if you can measure everything, then what you can do is understand your fitness landscape much more deeply. And instead of just waiting to see what comes out of your selection, in the future, you'll be able to design, because you'll have that whole landscape now measured. You'll be able to just go right to the spot that you want to pick the function that you want. So it is on bioRxiv. Feel free to take a look at that work. Another thing we're doing is trying to do a better job of measuring transcription in engineered cells. So this is important because transcription is used to measure and optimize the performance of engineered organisms. It's kind of the, in electrical engineering, you have current. In engineering biology, you've got, polymerase per second, as it were. And so here, this is part of our efforts to move away from relative measurements towards absolute measurements, numbers of things. And then I thought this was a small study that Drew Tack and others in my group did, but I thought it might be useful for us to spend a minute looking at. And basically what it says is if you precondition your host cells to the environment they're going to eventually live in, and then put in your engineered part, you get much better stability of your function. Kind of makes sense, but we wanted to check it. And we did this with an eye towards maybe making some cellular reference materials in the future. Okay, but what about building cells? Because Kate's gonna be unhappy if I don't talk about building cells, and I'm sure you will be too. So in my mind, NIST and my group, we live in the middle of this slide, this metrology slide. And this slide kind of summarizes what I see as the big challenges broadly for engineering biology in the future. Really in the near term, but maybe also not so near term. And if you look at the bottom left-hand corner, you see that minimal cells makes an appearance. And it's for this reason that I'm personally interested in minimal and synthetic cells. I think that they help us understand what's possible. They give us platforms for breadboarding function. And if we have a suite of these, all the way, if you imagine a spectrum from totally inert, non-living, chemistry to biology, like a full mammalian cell, when you're engineering the solution to a problem, you might not want to be constrained to just pick between those two, right? You might want to be anywhere along this axis. And if we can populate that axis with living systems or pseudo-living systems, and this is where I think build-a-cell can do a lot, then we can just, choose only as much complexity as we need, only as much functionality as we need to solve our problem. This is a little scary to talk about too, but I think part of this is going to necessitate building methods for gain of function. And because that's, building a synthetic cell is almost like the ultimate gain of function experiment. And I'm a physicist by training, so I'd be remiss if I didn't quote Feynman, what I cannot create, I do not understand. So that's where synthetic cells fall for me. And then my personal overly simplistic and extremely biased view of building cells is this. You have your primordial soup, you add a lot of great measurements and uncertainty analysis, and then that gives us a fighting chance of achieving our moonshot. And this lets you characterize identity and function, understand what more is possible, do some prediction design, to maybe even build something entirely new. And then of course, once you've built something, you want to be able to verify its actual composition, function, safety, and performance. And for all of these things, you need good measurements. So Jane is going to talk more about our Cellfree program, where we're focused on measurement infrastructure and specifically reproducibility and automation for this. So we had a workshop, and that report is available to you, where we talk a lot about folks' thoughts on where better measurements are needed. We are automating our Cellfree workflows, so reach out to us if you have any questions about that. Stay tuned for some publications coming out in the near term, and of course, we're happy to share preprints. In particular, right now we're examining the effects of DNA template preparation methods on protein expression of your Cellfree system. We're also talking with folks about reagent kits for cell-free. You know, people have this assumption that cell-based kits for cell-based workflows, they just assume that they're going to work in the same way for your cell-free system, but that's not necessarily the case for a variety of reasons, not least of which is that cell-based workflows, you know, you have kind of a built-in selection or purification step, right? Like your cell grew or it didn't. And so in the case of the DNA template preparation, you know that DNA was functional. But for cell-free systems, you're just all of that garbage that goes through your kit, and that's in your solution with your prepared DNA template, that's all entering your final reaction. So what's that doing to your yield? And we don't really know yet. We're also thinking about how we can integrate cell-free systems into an automated design, build, test, learn workflow for rapid genetic sensor development. Not clear there's a real role there yet, but we're thinking about that, and I'm happy to hear your thoughts about that as well. We're also involved in the Build a Cell Interlab and Integration Working Group. Again, Jane will talk more about that. Right now, you can see this great diagram I stole off the Google Drive for the group. There are lots of arrows going around, and those represent people shipping materials. Because right now, we're not really integrating parts yet, so much as testing parts in different laboratories to see how the separate parts work. But that's been really fun and interesting. And of course, we're finishing up some work with John Glass and company on minimal cells. I believe that others are looking at creating potentially a cell-free system from genomically minimal cells. I do point out that John gave an excellent Build a Cell seminar on this, incredible scientific journey towards a minimal cell, which I wouldn't even do it any justice. I was just along for a part of the ride. Okay, so I'm going to stop there talking about my group. Are there any questions about my group before I go into the portion of my talk on unsolicited advice? I'm looking at the chat. Feel free to speak up too. I don't mind being. Yeah, so John Pearson was asking about metabolic parameters and specifically in a minimal cell. And people are doing metabolomics. People are doing various omics on minimal cells. Some papers have come out on that. And then for cell-free, there are folks talking about, and you know what, Build a Cell has been talking about this too, but how do you develop something like an array of assays or a kind of a standardized well plate where each well is a different assay on your cell-free reaction or cell-free reagents. And then you would have that data as kind of it would be measurement assurance for you to see, is my system performing the way I expect? Is something weird today? Or did it perform in my hands the same way as it performed in somebody else's hands? And if not, can we see if our measurement plates agreed or not, right? Just looking at the chat. People want to keep holding pipettes. Great. And you know what? There will always be a place for people to do manual workflows, no doubt. And this goes back actually to our conversation at the beginning of this meeting today, Kate, where we and others were talking about, well, what's worth automating? And not everything is. And there's definitely a place for the quick and dirty benchtop experiment to de-risk something before investing a lot of time and effort and money, really, because you go through consumables like you wouldn't believe with these automation systems. They're monsters. You're like shoveling pipette tips in their maws. Okay, so moving on, Kate, this section's for you. It's actually for anybody who hasn't seen this stuff before. And I want to show it because I wish I had seen this stuff when I was a graduate student. So the first thing I want to talk about is the Helmeier catechism. So if you want to scope a project or write a proposal, this is not the only way to approach doing that, but this is a very proven and effective way of doing that. So if you want to know more about it, please follow the links. Please feel free to talk to me. Or talk to folks in your network who are mentoring you to see if they can help you interpret this. The other thing I want to also show you is this is a version of what you might hear referred to as a quad chart. So once you have your project that you want to propose, what do you take to a funder? What do you take to your PI? How do you sort of show in a snapshot what your project is about? And so feel free to take this template, do what you like with it. It's basically the Helmeier questions, but in a different form. And this is the project template that I use with my own group that I developed after I came back from DARPA. Any questions on this? And if it's useful, great. If it's not, sorry for wasting your time. The last thing I want to talk about with regard to unsolicited life advice is orienting yourself on the path. So I often get questions from people, how do I decide what to do next? How do I scope a project? And how is that different from a program? Or, what does it all mean? What am I doing with my life? Oh God, why did I get a PhD? I mean, we've all been there, and we've all been parts of these conversations. What I submit to you is that we all start in a place of agency. So we're empowered, and we like things. We have desires without reason, right? We're moved by things, and those come out in our interests. We leverage our interests to some kind of advantage. And then eventually, if we work really hard and we're lucky, then we might see some success. And I think we've all been, at various times at different places along that hierarchy. And we've all had a lot of advantage and a lot of success. Otherwise, we wouldn't have been this educated. We wouldn't have had the opportunities that we have to even just sit together like this in this conversation. But what I really want to push you towards is this last piece, which is worth. And I can't emphasize this enough, right? Because you can stand at success, but unless you want to die unhappy at in your cul-de-sac home, having an endless midlife crisis, please don't do that. I mean, do that. There's nothing wrong with that, okay? I mean, do that if that's what you want, but make it a choice, right? If you can take one more step over to worth, now you can have impact. Now you can have a chance at greatness. Now you can start saying things like, so success is, did I do it or did I not do it, right? Was I successful? But worth is, was it worth it, right? So how do you take your successes and your failures and roll them into worth? Then you can ask yourself, what questions are worth asking? What are worth answering? Are you the right person to answer that question? Or who do you need to bring along with you? You could ask yourself, how do I find worth in my liabilities to make them into assets so that I can take them from success and failure over to worth? And now you can start asking, what impact do I want to have? And what are you willing to do about it? So, et cetera, right? There's lots of variations on these and other questions you could ask as well. So when you're thinking about what to do next, or even whether what you're doing now is worth it, I'm just offering this as one framework for taking a closer look at that. Any questions? I know this is kind of weird and not usual for a scientific seminar. I don't mean to get all like new agey self-helpy here, but a lot of people have asked, so I wanted to throw this in. Okay, so I would be remiss if I didn't mention some further NIST resources. There's Genome in a Bottle, and there's also the Gene Editing Consortium. Fabulous people run these. If you do find yourself wanting to know about best practices or measurements for either of these, for sequencing DNA or for gene editing, I'm happy to hook you up, or you can follow the links. And I want to ask you also, what consortia, if any, do we need around enabling technologies and measurements for synthetic cells, or for some workflows, or for some systems that we think are critical for eventually making synthetic cells? So I hope this was interesting. I hope that there was at least something of value in this for everybody. I know that it's always fun for me to present my group, and hopefully we can strike up some friendships, strike up some conversations, strike up some collaborations. And I think that's what Build a Cell is all about right now. And I also warmly invite you to forward or apply for our postdoc opportunities, again, in machine learning. And that's part of analyzing that directed evolution, sort of high throughput data that's coming off of our BioFoundry, and also working that into algorithmically driving the BioFoundry. And then separately, there's a position open just broadly around synthetic biology, wet lab experience, hopefully towards genetic sensors. But we also have some needs around cell-free systems and yeast. There's lots of opportunity there. Thank you. So at this point, I do want to open it up for questions. And I'm happy to stay on for as long as people like.
Thank you so much, Elizabeth. I think there's a couple of questions in the chat.
What's the biggest bottleneck for measurements right now? Not enough fluorescent channels, right? So I think that the issue around, crosstalk and fluorescent channels is, it's obviously present in any fluorescent measurement. I don't know that that's our biggest bottleneck. Peter, can you be more specific? Are you talking about measurement like in biology writ large, or a subset of that?
You can unmute yourself too.
Large, wow. I might argue that it's actually cultural. I think that not enough people learn statistics and learn design of experiments and learn how to make a measurement. And that's one of the things I'm trying to fix with this seminar. But I didn't know it either. I started out in experimental high energy physics, where culturally the first thing that you do is they hand you a stack of statistics material to go learn. And they don't let you do anything until you've proven that you've worked through it. Maybe biology needs something like that too.
I just want to add to that question too, because I'm a chemist by training, and I'm used to the fact that when I do an experiment, I know where my molecules go. And now I'm pretending to do biology, and I don't know where my molecules go anymore. Elizabeth, do you think that will ever be possible to actually get chemical resolution, like we know where every single thing goes, or cells are just inherently too complex for that?
Well, there are multispectral techniques that can give you a lot of that information. I think that the challenge is, how can you know without destroying your cell? So yeah. If we can, I think that there's a lot of room for measurements, or like single molecule type measurements in living cells, plus maybe some fluidics and some microscopy. Now we still have the fluorescence channel problem. I'm not sure what to do about that. It's been an incredible tool. There are other, we can use magnetism, we can use all kinds of things that aren't fluorescence based, but I don't know that the tools are really that readily available right now. They're definitely very niche.
So there's not such a thing like a non-invasive mass spec for the whole cell?
No, that would be, I'm not sure how that would work. I think that Drew Endy and others who are working on cryo-EM are trying to address this. I mean, obviously it's cryo-EM, so your cell is dead, but yeah, it's a step in the right direction, I think.
Yeah.
So we're actually trying to go the other direction. We have a lot of single molecule expertise, but what we're finding is that what people have in their labs is they don't have single molecule fluorescence microscopes, right? They've got flow cytometers. So how do we get as much, how do we understand the limits of the information we get off of our flow cytometer, right? And we can do the hard measurements at NIST to calibrate then, and verify what we think we can say meaningfully based on our flow measurements, right? So that's the kind of stuff that we can work on. Okay, I know there's lots of chat here. So how many parameters do you measure with your Measure Everything project, and with what time sampling? Great question. So the Measure Everything project in this case is you have a library that you're building of 10 to the 6 or however big your library is of different versions of genetic sensors. And so what we're measuring is the fitness of each one of those sensors. and we do it over time. So because we measure full growth curves. So what you'll notice about our BioFoundry is that we don't have a big bank that's just a rack of plates that are incubating, because we actually incubate our plates on our plate readers so that we can measure their growth curves of the whole populations the whole time. And what we can do then is using DNA barcoding, understand who was where and how they were growing. So we can tie that fitness measurement to the function of that sensor that we're developing. So that's the key. I see calls for standardizing. So liposome standardizing. So Kate, do you want to talk about this? Because this is actually a protocol that you guys put out, right?
I do want to standardize liposome formation. So Build a Cell actually did put out a sort of standardized protocol, a GitHub protocol where people were putting comments for about 6 months and trying to reproduce that protocol and keep improving on it. What's standardized is the method of the production of the liposomes themselves. We don't have a standardized method for measuring encapsulation rates, especially encapsulation rates with a single liposome resolution. Because right now, we can measure encapsulation in bulk by measuring bulk fluorescence of whatever is encapsulated, but we don't know what's the distribution of that between liposomes. And if you look at some microfluidics data, you can see that that's actually not stochastic. You have some liposomes that are really stuffed and some that are pretty empty.
My husband actually has a measurement platform based on nanofluidics that I think is a little bit small for the liposomes that you're making, but it could absolutely be sized up. So that might be worth a conversation worth having. His devices are based on a fluidic staircase design, but he does mold them out of PDMS now. So they might be something that would be more available for people and more usable than a glass fluidic device.
Yeah, that would be really useful, especially if we want to grow this field and get into business of making money and start making drugs based on this technology. We need a way to standardize our samples beyond just preparation. We need to have a way to measure if your end product, if your liposomes that encapsulate TXTL actually have the right amounts in the right proportions of everything.
Right, because if you could sort of wick in a representative volume, a representative sample of your encapsulated liposomes, then you'd be able to do a size distribution instead of doing just a DLS or other kind of measurement.
Yeah, a size distribution, but also an distribution of stuffing, like what
That's
percentage of your lip. What percentage of liposomes have stuff that you were interested in versus what percentage are duds?
Well, I mean, couldn't flow cytometry work for that? If you could optimize the flow protocol?
If you have a fluorescent marker, then sure.
Right.
A lot of the time when you think about making therapeutic liposomes that, let's say, produce a protein that has some activity against certain cancers, they might not be fluorescently labeled.
Sure, but you could at least prove out your workflow.
That's true, yeah.
All right, 6 minutes. Anyone else want in? Tell me what you think. Questions, comments, complaints, concerns. I'll take commendations as well.
I think everyone's just wrapping their heads around the fact that we're going to be replaced by robots soon.
Well, but that's not true entirely, right? Because robots can't ask if it was worth it yet. Unless they can, then we're really in trouble.
Yeah. I just want to say I really appreciate that you actually went into this kind of details of why we're doing it, not just how we're doing it.
I think it's always good to remind ourselves like what it is we're trying to do, because I think we get so in the weeds of our technical work that it's hard to pick out, especially now. God, I mean, everybody's living the pandemic life, right? So picking your head up from that is hard, but it's worth it, because otherwise, you can't see where you've been and you can't see where you need to go.
Yeah. Okay, so if there's no more questions, I want to say thank you very, very much, Elizabeth, again.
You are most welcome. And I will send my slides to Kate so she can post them. And feel free to do with them as you like.
Great. Thank you very much. And thanks, everyone. And you can always email Elizabeth. Please email. Please bother her.
Please do. We're from the government. We're here to help.
All right. Bye, everyone. And bye, Elizabeth.
Bye, everyone.
Thank you.